Welcome to Dr. Shankar Golapudi's Neuroscience class. In this video, we will discuss neuroanatomy and brain structure, neurotransmitters and synaptic signaling, neurophysiology and action potentials, cognitive neuroscience and brain imaging techniques, neurological disorders and diseases, neurodevelopment and neuroplasticity, neuropharmacology and computational neuroscience. Neuroanatomy and brain structure are fundamental aspects of neuroscience. Let's delve into some key components. First, the major divisions of the brain: the forebrain, or prosencephalon, the midbrain, or mesencephalon, and the hindbrain, or rhombencephalon. Next, the cerebral cortex, which includes the four lobes: frontal, parietal, temporal, and occipital. Key areas in the cortex are the motor cortex, somatosensory cortex, visual cortex, and auditory cortex, along with Brodmann areas. Then we have subcortical structures such as the basal ganglia, which includes the striatum and globus pallidus, the thalamus and hypothalamus, the amygdala and hippocampus, and the cerebellum. Moving on to the brainstem, it consists of the midbrain, pons, and medulla oblongata. The spinal cord includes grey matter and white matter, as well as spinal nerves and nerve roots. Cellular components are next, with neurons like pyramidal cells and interneurons, and glial cells such as astrocytes, oligodendrocytes, and microglia. White matter tracts include the corpus callosum, internal capsule, and spinal tracts like the corticospinal and spinothalamic tracts. The ventricular system consists of four ventricles and cerebrospinal fluid circulation. The meninges include the dura mater, arachnoid mater, and pia mater. Finally, the vascular supply involves the circle of Willis and major cerebral arteries. The cerebral cortex is the outermost layer of the brain and plays a crucial role in higher-order cognitive functions. Let's explore it in more detail. The structure consists of grey matter, primarily neuronal cell bodies, and is 2 to 4 millimeters thick with a highly folded surface of gyri and sulci to increase surface area. It is divided into 6 layers, each with distinct cell types and connections. The 4 major lobes are the frontal lobe, which handles executive functions, planning, decision-making, and motor control, with key areas like the prefrontal cortex, primary motor cortex, and Broca's area. The parietal lobe is responsible for sensory integration, spatial awareness, and navigation, with key areas like the primary somatosensory cortex and superior parietal lobule. The temporal lobe manages auditory processing, memory formation, and emotion, with areas like the primary auditory cortex, Wernicke's area, and hippocampus. The occipital lobe handles visual processing, with the primary visual cortex being a key area. Functional areas include primary sensory areas that receive direct sensory input, association areas that integrate information from multiple sensory modalities, and motor areas that plan and execute movements. Brodmann areas provide a numerical classification of cortical regions based on cytoarchitecture, with examples like Area 17 for the primary visual cortex and Areas 44 and 45 for Broca's area. Lateralization refers to certain functions being more dominant in one hemisphere, such as language typically being lateralized to the left hemisphere in most right-handed individuals. Plasticity is the cortex's ability to reorganize in response to experience or injury, crucial for learning, memory, and recovery from brain damage. Connections are extensive between different cortical areas and with subcortical structures via white matter tracts. Cortical columns are vertically organized neurons with similar response properties, important for information processing within the cortex. Neurotransmitter systems, including glutamate, GABA, and dopamine, influence cortical function. Development follows an inside-out pattern during embryonic development and continues to mature into early adulthood. Certainly, neurotransmitters are chemical messengers that facilitate communication between neurons in the nervous system. Let's delve into the topic. The major classes of neurotransmitters include amino acids like glutamate, which is excitatory, GABA, which is inhibitory, and glycine, which is also inhibitory. Monoamines include dopamine, serotonin, norepinephrine, epinephrine, and histamine. The cholinergic class includes acetylcholine, while peptides include endorphins, substance P, oxytocin, and vasopressin. Synthesis and storage involve production in the neuron's cell body or axon terminals and storage in synaptic vesicles. The release mechanism is calcium-dependent exocytosis in response to action potentials. Receptor types include ionotropic receptors, which are ligand-gated ion channels, and metabotropic receptors, which are G-protein-coupled receptors. Termination of action involves reuptake by transporters, enzymatic degradation, and diffusion. Functions of neurotransmitters include excitation or inhibition of postsynaptic neurons, modulation of neural circuits, and regulation of mood, cognition, and behavior. Neurotransmitter systems include the glutamatergic system, which is the primary excitatory neurotransmitter in the central nervous system and is crucial for learning and memory, such as long-term potentiation. The GABAergic system is the primary inhibitory neurotransmitter in the central nervous system and is important for anxiety regulation and sleep. The dopaminergic system is involved in reward and motivation, motor control, and is implicated in Parkinson's disease and schizophrenia. The serotonergic system regulates mood and the sleep-wake cycle and is the target of many antidepressants like SSRIs. The cholinergic system is involved in attention and arousal, memory formation, and neuromuscular junction signaling. Neurotransmitter imbalances are associated with various neurological and psychiatric disorders, such as depression, anxiety, schizophrenia, and Parkinson's disease. Pharmacological interventions target neurotransmitter systems, with examples including SSRIs, benzodiazepines, and antipsychotics. Co-transmission refers to neurons releasing multiple neurotransmitters, allowing for more complex signaling and modulation. Neuromodulators are substances that can alter the effects of neurotransmitters, with examples including adenosine and nitric oxide. Let's dive deeper into GABA, or gamma-aminobutyric acid, the primary inhibitory neurotransmitter in the central nervous system. First, let's talk about its chemical structure and synthesis. GABA is a 4-carbon non-protein amino acid. It's synthesized from glutamate by the enzyme glutamate decarboxylase, or GAD, and requires vitamin B6, also known as pyridoxal phosphate, as a cofactor. Now, regarding its distribution, GABA is found throughout the central nervous system, especially in the cortex, hippocampus, basal ganglia, and cerebellum. It's also present in some peripheral tissues. Moving on to GABA receptors. GABA-A receptors are ionotropic, meaning they are ligand-gated chloride channels. They have a pentameric structure with various subunit compositions and are the primary target for benzodiazepines, barbiturates, and some anesthetics. GABA-B receptors, on the other hand, are metabotropic or G-protein coupled. They activate potassium channels and inhibit calcium channels, leading to slower, longer-lasting effects compared to GABA-A receptors. GABA-C receptors, now classified as a subtype of GABA-A, are ionotropic but have distinct pharmacology. They are primarily found in the retina. As for its functions, GABA reduces neuronal excitability throughout the nervous system. It plays a crucial role in maintaining the excitation-inhibition balance and is involved in anxiety regulation, sleep, and muscle relaxation. It's also important for neural development and plasticity. In terms of metabolism, GABA is removed from the synaptic cleft by GABA transporters, or GATs, and is metabolized by GABA transaminase, or GABA-T, to succinic semialdehyde. GABAergic neurons include interneurons in various brain regions, Purkinje cells in the cerebellum, and neurons in the striatum and substantia nigra. Clinically, the GABA system is a target for anxiolytic drugs in anxiety disorders, anti-epileptic drugs in epilepsy, and sleep medications in insomnia. Alcohol enhances GABA effects, contributing to its sedative properties. Pharmacological agents affecting GABA include benzodiazepines like diazepam, which are positive allosteric modulators of GABA-A receptors, barbiturates, gabapentin, pregabalin, and vigabatrin. GABA may also act as a neurotrophic factor, playing a role in neuronal development and differentiation. Interestingly, GABA is found in non-neural tissues like the pancreas, where it may regulate insulin secretion, and the immune system, where it might modulate immune responses. GABAergic signaling in the suprachiasmatic nucleus helps regulate circadian rhythms. Finally, GABA can modulate the release of other neurotransmitters like dopamine and serotonin. GABA receptors and their subtypes. Let's start with GABA-A receptors. These are ionotropic or ligand-gated chloride channels with a pentameric structure composed of various subunit combinations, including alpha, beta, gamma, delta, epsilon, theta, pi, and rho. The most common subtype is composed of 2 alpha, 2 beta, and 1 gamma subunit. GABA-A receptors are fast-acting, operating on a millisecond timescale. They increase chloride conductance, typically leading to hyperpolarization. They have several binding sites, including the GABA binding site between alpha and beta subunits, the benzodiazepine binding site between alpha and gamma subunits, and sites for barbiturates and neurosteroids. These receptors are modulated by various compounds, including alcohol and anesthetics. GABA-B receptors are metabotropic or G-protein coupled and are composed of 2 subunits, GABA-B1 and GABA-B2. They are slower acting, with effects lasting from seconds to minutes. These receptors activate G-protein coupled inwardly rectifying potassium channels, inhibit voltage-gated calcium channels, and inhibit adenylyl cyclase. GABA-B receptors are found both pre- and postsynaptically. Presynaptically, they inhibit neurotransmitter release, while postsynaptically, they produce slow inhibitory postsynaptic potentials, or IPSPs. GABA-C receptors, now classified as RHO subunit containing GABA-A receptors, are ionotropic and composed exclusively of RHO subunits. They are primarily found in the retina and are more sensitive to GABA than GABA-A receptors. They are resistant to bicucaline, a GABA-A antagonist, and baclofen, a GABA-B agonist. GABA-C receptors also exhibit slower desensitization compared to GABA-A receptors. Clinical applications of GABA-related drugs are quite extensive and varied. In anxiety disorders, benzodiazepines such as diazepam and alprazolam enhance GABA-A receptor function. Buspirone, a partial agonist at serotonin receptors, indirectly affects GABA systems. For epilepsy, gabapentin and pregabalin increase GABA synthesis and release, while vigabatrin inhibits GABA transaminase, increasing GABA levels. Tiagabine acts as a GABA reuptake inhibitor. In the treatment of insomnia, drugs like zolpidem and zaleplon are GABA-A receptor agonists selective for the alpha-1 subunit. Esopiclone is a non-benzodiazepine GABA-A receptor agonist. For muscle relaxation, baclofen, a GABA-B receptor agonist, is used to treat spasticity. In anesthesia, propofol enhances GABA-A receptor function. For alcohol use disorder, acamprosate is thought to modulate both GABA and glutamate systems. Lastly, in the management of neuropathic pain, gabapentin and pregabalin are also used for certain types of chronic pain. GABA's role in specific brain functions is, well, quite extensive. Let's break it down. First, anxiety regulation. GABAergic transmission in the amygdala and prefrontal cortex modulates anxiety responses. Next, sleep GABA. In the hypothalamus and brainstem helps regulate sleep-wake cycles. When it comes to memory and learning, GABAergic interneurons in the hippocampus play a crucial role in memory formation and retrieval. Motor control is another key function. GABA in the basal ganglia and cerebellum helps coordinate movement. For sensory processing, GABAergic inhibition shapes sensory responses in various cortical areas. Circadian rhythms. GABA signaling in the suprachiasmatic nucleus regulates circadian cycles. And finally, neuroplasticity. GABA influences critical periods in development and ongoing plasticity in the adult brain. Now, let's dive into the metabolism and synthesis of GABA. Synthesis. GABA is primarily synthesized from glutamate by the enzyme glutamate decarboxylase, or GAD. There are two isoforms of GAD, GAD65 and GAD67. This process requires vitamin B6, also known as pyridoxal phosphate, as a cofactor. Storage. Once synthesized, GABA is stored in synaptic vesicles by the vesicular GABA transporter, or VGAT. Release. GABA is released through calcium-dependent exocytosis in response to action potentials. Reuptake. GABA is removed from the synaptic cleft by GABA transporters, or GATs. There are four subtypes: GAT1, GAT2, GAT3, and BGT1. Metabolism. Finally, GABA is metabolized by GABA transaminase, or GABA-T, to succinic semialdehyde. Succinic semialdehyde is then oxidized to succinate, which enters the Krebs cycle. GABA's interactions with other neurotransmitter systems are, well, quite fascinating. Starting with glutamate, GABA and glutamate often have opposing effects, maintaining an excitation-inhibition balance. Interestingly, glutamate is the precursor for GABA synthesis. Dopamine. GABA can modulate dopamine release in the mesolimbic pathway, which is important for reward processing and motivation. Serotonin. The GABAergic and serotonergic systems interact in mood regulation. Some serotonergic drugs like SSRIs may indirectly affect GABA function. Norepinephrine. GABA can influence noradrenergic transmission, affecting arousal and attention. Acetylcholine. GABAergic interneurons in the cortex can modulate cholinergic signaling, impacting attention and memory. Endocannabinoids. Endocannabinoids can modulate GABA release, providing a form of retrograde signaling. This comprehensive overview covers the major aspects of GABA in neuroscience. Thank you for listening.